So it's time, and we should start now. And my name is Minoru Go from Department of Systems Medicine. And thank you very much for coming to the third lecture of the Centennial lecture series of Keio University School of Medicine. So this is the third time, and every last two lectures, I gave a pretty lengthy introduction. So I'm going to skip this now. Okay, this is just showing you why we are here and why we are celebrating these occasions. And let me move on directly to introduce Dr. Schlessinger. It's my great honor and pleasure to have Dr. David Schlessinger as a speaker today. He has had a remarkable career, and that's one of the titles showing that more than 60 years career in molecular biology. And just to give you illustrate some example, he started to do experiment when he was an undergraduate student at the University of Chicago. And then he moved on to Harvard for PhD. And actually he was the first graduate student of Jim Watson, who discovered the DNA structure. And then he moved on to, after PhD, he moved on to Paris and went to work with Jacques Monod, who also got a Nobel Prize for seminal discoveries. And so David worked there for 2 years and came back to the U.S. and became assistant professor at the Washington University, St. Louis. And since then, he was there for 35 years as a professor and then also director of the Genome Center. And the Genome Center David opened there was the first Genome Center, period. And he indeed started and then started a model of the Human Genome Center, which basically spread to many, many places. And David, of course, Dr. Schlessinger, of course, made seminal contributions on initially X chromosome analysis of the Human Genome Centers. And then he decided to move to NIH, and that was 1997. And since then, he's been there for, I think, close to 20 years now. And of course, he started something new, and then also there are many, many things that he's been discussing during this lecture series. But so I'm not going into too much details of those. And but just one thing that I think I wanted to talk a little bit about is that I was with him today, this afternoon, and we were just talking, and then he started to talk about this discovery of course, the reverse transcriptase and blah, 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 and then Howard Temins and David Baltimore's, and all of those are actually David somehow involved in those and was friends. And then, I think David really indeed has been in central and in center of the most of the molecular biology that you you've learned in the textbook and type of things. And of course, David himself made really a tremendous contributions to the field. So today, this third lecture, Dr. Schlessinger is going to talk about somewhat the historical aspect of the molecular biology, which will be intertwined with his own research project he started very early on. And he said that he would save the most important and interesting things to the last. So you guys should stay until the end. Okay. Well, that's it. So Dr. Schlessinger, please.
Thank you, Minoru. It's a great pleasure to be with you on the occasion of this centennial celebration of the remarkable achievements of Keio medicine. And I think the organizers may have tried to look for someone and someone's research that went as far back in the 100-year history as possible. And in fact, today I will describe the now completion of an experiment that did start 60 years ago. One of the reasons for doing that, beyond explaining the history of how it developed, is to show you the way in which what was possible depended on the technology at the time. You may hear people often say that in a particular field, it was amazing that there was just-in-time development of a technique that was needed. But in fact, it's the reverse. It's because the technique is developed that the experiment becomes possible. And I'll show you that both with respect to particularly the ribosomal DNA and to as the Genome Project developed. It's true that this will be historical and it'll take some time. But if you doze off or lose interest, if you'll bear with us, there will be results at the end that I think you'll probably find interesting. So this begins with the first work I did as a graduate student. When I entered the nascent laboratory of Jim Watson in 1957, he had a large office with a desk lamp, and on the desk lamp there was a little Scotch tape piece of paper which said DNA, RNA, protein. That was the model that they were attempting to demonstrate and figure out. At the time, there was a visit from Francis Crick, and Crick gave a talk in which he gave his postulates, which was that if you want to understand biological function, study structure. At the time, there was already some evidence about ribonucleoprotein particles, what we now call ribosomes, and that they were somehow involved in protein synthesis. So I took on the project of beginning to look at the structure. The first job I had was to determine the size and composition of the ribosomal particles. And the first slide is the least readable of all the ones you'll see. It deals with the physical measure of molecular weights of the ribosomes. I found a way by differential centrifugation to get pure preparations of the ribosomal subunits and the 70S ribosomes from E. coli. And having achieved that, I could use physical chemistry to measure their molecular weights. It was a combination of the diffusion constant, the rate at which they diffused in a solution, their sedimentation rate, and the intrinsic viscosity of the solution. That permits you to get two different estimates of molecular weight, and the numbers are unimportant. They happen to coincide very closely to the numbers that were seen later, even by crystallography. Physical chemistry is a very good way to operate on biological systems when it's possible. There were a number of steps involved in ribosome formation and function that occupied me for a number of years. And this was one of the discoveries. We knew that bacterial ribosomes contained the two ribosomal RNAs, the 16S and 23S. But we found that in a mutant of E. coli lacking a particular RNase, RNase 3, we could spot transiently a very large transcript, a 30S transcript, which contained both of the mature RNAs. So the bacterial cells, like human cells, ensure equal numbers of the ribosomal RNAs by making them in a single precursor. And what this suggested was that this RNA would be cleaved by RNase 3, and that proved to be the case. The two mature RNAs are enclosed by double-stranded stems, and RNase 3 was already known to cleave up double-stranded DNA bits, or double-stranded RNA, but it turned out that it could also cut double-stranded RNA segments within an otherwise single-stranded RNA. So the basis of this was the first RNA processing step to be discovered, and it was based on Watson-Crick base pairing, as have been nearly all the processing signals since. So it was a simple rule that developed to be practically universal. For most of a decade, what we were doing was to establish what I called the ribosome cycle in protein synthesis. And the idea was simple. We could show that the ribosomal RNA in proteins were made. They went through a series of precursors to form the subunits. And these would then join to a messenger RNA to form a protein chain from amino acids, and then the message after a while would be degraded. At first, the notion had been that a ribosome was formed as a 70S, and it moved as such from one message to another. But it turned out that, in fact, the subunits were always in a free pool, and periodically a couple would form from a 30S and 50S to a 70S. The additional idea that then came up was the notion that a single message could service more than one ribosome at a time, the notion of a polyribosome. So you could start protein synthesis with one ribosome, it would get out of the way, and another one would join the initiation site. So you'd have a number of ribosomes functioning at the same time. This was an idea, but no one had seen them. Because if you broke open cells by typical means, there'd be shearing forces, and all you would see would be the 70S, or in mammalian cells, the 80S ribosomes. And we overcame that problem by a technical development, a simple one, where we made mutants of E. coli that grew in a fragile form. They were sucrose dependent and grew well then. If we removed the sucrose, they lysed. And the easy lysis permitted us to actually see polyribosomes for the first time. This was the then new technique of zonal sedimentation and sucrose gradients, still useful in many cases. And you can see here clearly the peaks of 30S and 50S ribosomes, the 70S monomers, dimers, trimers, and so on. The notion of the polyribosomes enabled us to begin to analyze the effects of antibiotics that affect protein synthesis. A great many of these act at the ribosome level, and we were able to pinpoint the points at which they block the ribosome action. The most interesting of these, for example, was streptomycin. And you can see here some data about that. So again, in a wild-type E. coli, you get the spread of the polyribosomes that's normal. If you add streptomycin, ribosomes that are on a message can limp to the ends of the message slowly. But when they try to reinitiate, they form an aberrant initiation complex. You can get nearly to the first peptide bond, but that's difficult. And the ribosomes accumulate as dead 70S particles. So you kill the cell by killing the ribosomes. Around this time, it was already possible to begin studying human ribosomes as well. And this became a great focus of work. There was the first observation of the mammalian polyribosomes in extracts of reticulocytes. The labs of Muramatsu and Tijin realized that polymerase I was the specific one for ribosomal RNA, and they defined the features of the promoter sequence. And we even began to look at the effects of methylation of ribosomal RNA on how processing goes in mammalian cells. One of the most extensive studies we did at the time was that by the beginning of the 1980s, we could define the processing pathway from what was the large ribosomal precursor RNA, again including the two major ribosomal RNAs, mature RNAs, to the final products. And we defined several processing pathways, which actually have been recently reestablished and confirmed after a number of decades. I just want to point out that at this point, there was no sequencing information. And so the technology consisted in a very elaborate procedure of using refined restriction maps and what was called S1 nuclease mapping, now an obsolete technique, but was capable of defining the approximate ends of all the precursors and the time course of what was happening. Now, in the intervening years, you should all know the continuation of ribosome research. It was a succession of triumphs. The structure and the ribosomal RNA action protein synthesis were determined with the crystallography and the brilliant work of Noller's group on the actual function of ribosomal RNA. And at a certain point in the 90s, there was actually the sequence of the human rDNA encoding the 45S pre-RNA that was assembled from a bunch of small clones. A number of groups, including ours, was involved, but the critical work was done by Gonzalez and Sylvester. And they published a sequence of what would be a coding unit of human rDNA, which is still the reference sequence that's in the genome browsers. But the study of rDNA essentially then stopped. The field stopped. There was no further study of ribosomal DNA and remarkably little about ribosomes. Why? We knew that there were 400 tandem repeats of ribosomal DNA, roughly. They're distributed on the short arms of the 5 acrocentric pairs of human chromosomes. So 13, 14, 15, 21, and 22. But the general idea was that the repeat units were boringly the same. Once you had a sequence of a unit, why bother to look at further at the analysis of the rDNA? Well, I was thinking at the time, could you really have 400 copies that were identical? If you think of Susumu Ono's famous work, he postulates that, and it's now accepted widely, that evolution proceeds by duplicating genes and having them diverge. So were these really identical? And what else might be in the rDNA regions? Were there other genes there? If nobody looked at it, you wouldn't know. How do you set the rate of transcription of the ribosomal copies? There are a great many copies. No more than half of them are ever active at any one time. Well, we wanted to continue the studies, but there was a block in technology. The cloning systems available were inadequate to provide a full copy of rDNA intact. You just had little pieces that you had to fit together. And certainly you couldn't look across a region of multiple units. So there's no way to do it. At this point, we realized, this is 1986, after multiple tries to do this, that exactly the same technical block had to be faced for the growing interest in the possibility of the Human Genome Project. There was a committee of a group of us that began to discuss the possibility of analyzing the structure of the human genome. It was a much larger goal than the ribosomal DNA, but the block was the same. For selling this is one of the ways that I put it at the time. The full human genome sequence for biology will be like the periodic table of the elements for chemistry. Once known, it will be the starting point for biological research for the rest of time. So there's a lot of enthusiasm, but there was no way to do it. We only had these small clones, and by the way, no source of funding. This situation was changed by several technological developments in my colleague at the time, Maynard Olson's lab at Washington University in St. Louis. And they provided the first genome center and another step, by the way, toward analyzing ribosomal DNA. Olson's lab developed yeast artificial chromosomes as a cloning vector. And I'll show you some of the original work on that in a moment. This led us to have a logistical construct that we call the Center for Genetics and Medicine. And that was what Minor was referring to as the model genome center. And then we were able to get funding, which permitted us to actually start. These are now slides from Transparencies. This is 1986. These are the first slides made on the trial Mac program for making color slides at the time. And you'll see a number of these as we go along. The yeast community had assembled everything that you needed to make a chromosome. Telomere sequences, centromere sequences, and they could make a small artificial chromosome. What David Burke and Maynard's lab did was to cut this open and have a cloning site so that you could actually restrict a nucleus, treat human DNA or other DNAs, and clone fragments of large size between the two telomeric ends. So, in other words, they transformed a yeast artificial chromosome into a cloning vector of yeast artificial chromosomes. This led to the following formulation. So if you think about traditional genome analysis at the time, you had a map resolution in the 3 billion bases of the genome, which was limited to chromosomes, a little bit of information from linkage mapping, and then you had small clones. The largest available would be cosmids, which would come near 40 kb. And this was an immense gap. So the goal of any map, there's a theory of mapping. The goal is continuity, contiguity across the region you're trying to map, and no holes. That's what you wanted, and you couldn't come near that. So the principle was vertical integration of genome analysis, and the idea was to interpose these YACs, which could clone as much as a megabase or more of DNA, as an intermediate step, which could then be broken down further to reach, as we could in nematodes or other DNA, the level of sequence. An easy way of thinking about this is that we were faced with cosmids or lambdas with tens of thousands of pieces of DNA to try to fit together. Instead, with the YACs, it became much more like a child's jigsaw puzzle. Anybody could fit it together at that point. So Olson's formulation was to overlap the YACs to achieve long-range contiguity, reconstructing entire human chromosomes outside of the cell, and then that would be a step toward complete sequence analysis. At the time, sequencing was also just developing, so it wasn't ready, but at least we had an approach to getting the maps. Now, there were challenges to get to YAC-based vertical integration. We had to get the YACs. We didn't have a library. There were no large libraries of clones at that time. The YACs had to be stable and faithful to the sequence, and they actually had to be able to clone all of human DNA, which includes a lot of strange regions. So we had to make YACs, recover them, and overlap them. And again, this required technology. Since it wasn't available, we had to be able to be ready to adapt new techniques or to do them ourselves. And so that was the reason for the assembly for this center. And it was thinking in terms of a set of core facilities. One which would be involved in the mapping itself and would lead to sequencing, and two special cores, one for technical development and the other what we called information handling. The word bioinformatics wasn't there yet. Now these were new. Technical development groups were common in chemistry or physics. but they did not exist in biology. So the idea of having a group that would be adapting things and trying to create them was new, and there was no information handling at the time. We had to convince informatics people, computer scientists, to join us. So these were the critical cores. How did it proceed? I'll give you a feeling for it here. First, there was a local strategy. The idea was to do it at a medical school because the people there could interact and they would want the maps and the clones, for example. And then there was an international strategy. We chose the X chromosome as a prime target. For those of you who've had any background in medical genetics, you'll know that because the males only have one X chromosome, recessive mutations are exposed. So more than half of the known genetic pathology was on the X. So we'd get a high yield of information that would be valuable to the community. And there was, in fact, a large community of human geneticists. They had some clones, materials, and interests that would benefit both their work and the center. As for support, this never changes. We had the support locally. We got space. But funding. When we went to the NIH and asked them, we told them we had this cloning technology. We'd like to see if we could get maps. And I got the worst review of my career. It was one line. This is only mapping. There was a lot of opposition to this project. People wanted money to learn how to work on a disease. What was this for? So we went locally. In St. Louis, there's the McDonald Foundation, which gave us $1 million of startup. It's the same foundation that's funding the interactions between Keio and Washington University in St. Louis right now. And then the Monsanto company, which was the largest in the town and had a research unit, also gave me $1 million after a presentation. So we had enough money to start. Of course, when the center was successful, then the NIH created a center program that funded them. We assembled heads of the cores for the mapping facility, the technical development, the informatics and mapping. And I'll mention some of these people perhaps later. And then the staff for the project. There were some graduate students and postdoctoral fellows to help with technology development and mapping. And then we had collaborating groups at Baylor, particularly, that was interested in the X chromosome, and also many places around the world. We also started a second project for chromosome 7, which is about the same size as X, and it was run by Eric Green with Maynard. Eric is now the head of the Genome Institute. The bulk of the work, however, was done by undergraduate students, especially, and some technicians, because you had to create libraries of hundreds of thousands of clones and size them and get probes and do the map assembly. Undergraduates have a lot of energy. So at every step, technology was critical. You can only go ahead if you have a technique to do it. For example, how are we going to store 50,000 clones? There were two possibilities, to give you an idea, that we discussed. One was to make custom-made plastic so you get dense storage of clones. We opted instead for 96-well plates, which were already starting to be used by immunologists, because it would enable us to have storage that was easy to handle with a commercial product and easy to transmit to other locations. We wanted to be able to share the materials. How could we get all these yak clones together? We had to have a library, which hadn't been done before. How could we recover the overlapping yaks, and how would we store the information? And I'll just give you a capsule idea of how it went. First of all, how to get a library of clones. Well, yeast when you make artificial chromosomes, the yeast have to be made fragile in order to introduce the DNA. And then they're plated in a top agar, which is very gooey. A clone that has an insert is red. There's a gimmick genetically so that if any yak that has formed with a human DNA insert will be red. But the plate had a whole bunch of colonies. At first, we were picking the red ones one by one and repurifying them. Too slow, impossible. So a graduate student, Randy Little, came up with a solution. He took a block of wood and made a manifold of a very large number of pins, all drilled to the same level. This permitted him then to sample from the top of the plate, transfer it to a selective medium, and on the selective medium, only the clones with an insert would grow. So they were automatically purified. And then these could individually be picked up, grown up, stored, and analyzed further. Every one of the clones had to be sized. And again, there was a new technology. In this case, George Carl, another graduate student with Maynard Olson, developed pulse field gel electrophoresis, where you could visually see all of the normal yeast chromosomes and then the alternative new ones. These could then be transferred to film, where there'd be a signal from radioactive DNA probes from human DNA, a repetitive sequence that would light up all the clones. And here you see another plate where the yeast artificial chromosomes are clear, and you have yeast artificial chromosomes which reach well over 800 kb. So this is doing damage to the idea that you have to get coverage over a wide area. Here's a technician individually measuring all the clones. Then the clones would be, having been sized, would be grown up and stored on these 96 well, then 384 well plates. Now this was the most serious technological block. How do you find a particular yak clone that you want? Let's say you want to study factor VIII. We had little pieces of factor VIII from labs that were studying it, so we had a probe to start with. But we've got 50,000 clones. All we had was these probes, and the method, which you don't have to read, I'll just describe it quickly, was hybridization. So we had 50,000 clones, which we would stamp in tiny amounts on hundreds of filters, nylon filters. They would be lysed on the filters, and we would apply a red-hot solution that contained a highly P32 labeled probe. The probe would be allowed to incubate, and if there were any clones somewhere in the 50,000 that would hybridize, they would hybridize there. The material would be transferred to the film, and we would see a radioactive spot that lit up over background. If you think this sounds bizarre, it was. It was a lot of work. Imagine liters and liters of hot solution that you had to store until it decayed. But it was enough to start with. We calculated that we could only do 100 screenings a year, top speed, but it was enough to begin. And to simplify the initial mapping for the X, we had some pieces of DNA that had been isolated in the community, others that we made locally, the DX probes that we made from fragments of X chromosome DNA. And we had a series of cell hybrids that we could map to make sure we were getting the probes and clones going to the regions where these probes had lit up cytogenetically particular bands. The more probes we had in a region, the more clones, yak clones we had recovered, and the sum of the DNA began to be significant. So we could get clones. They seemed to be representing a large number of probes. And we then took one of the first ones for a gene that had been widely studied already, glucose-6-phosphate dehydrogenase. And we actually sequenced the yeast artificial chromosome that contained it and plasmids, which had the full gene, and showed that the sequence was identical. So this was the first evidence that the yaks were actually faithful to the genome sequence. In fact, we took a yak with this G6PD and transfected it into cells and showed that it made the protein and enzyme. Of course, hybridization was inadequate. It was slow and exhausting. And with all this radioactivity, very expensive to do. Everything was revolutionized by a technological development, PCR. PCR made the difference. Maynard did a formal representation of this in a paper in Science, and this is the critical sentence. You allow short DNA sequences to be detected easily with high specificity and sensitivity so that bits of sequence themselves become the landmarks on the physical map and can be used to recover the clones that will give you the complete map. At this point, Volker Novotny, who had considerable engineering talent, built an elaborate robotics station. It had room for at least 500 trays with 384 samples, and you could dial from a console the ability for an arm to pick out any of the plates and sample the clones you wanted to look at. The other half of this was a very clever optimization algorithm so that we could use pools of clones and with no more than 21 PCR reaction, identify the exact clones that gave a PCR signal with a specific primer pair. So now we were in business. We could do this, what was called sequence tagged site mapping of the X chromosome with essentially automated retrieval of YACs and PCR reactions automated through the action of undergraduate students. And this was the obvious flow chart. The technical development had given us robotics throughput. We made the STS primer pairs in a core, did the screening, produced the YACs, then used the project labs to help doing the mapping, assemble the maps along with the informatics core. And this would then give us contigs, overlapping clones, and finally move toward closure of the map. So you get a YAC STS map from this. More important, within a year, we had done well over 400 probes. And now I could do a test to see whether we were really going to get there. The question was all of the genome being represented? And there's a very appropriate mathematical formulation for this, the Poisson Distribution. We knew the size of all the clones, so we knew we were usually using a library that had 3 genomic equivalents. There was enough DNA to cover the genome 3 times. By the Poisson distribution, then an average probe would give us roughly 3 clones, but we could predict how many clones we should be getting with 1 to up to 10 clones, and how many would not find anything in a 3-hit library. And the actual data are the red bars. It was pretty close to the random expectation. There were more probes that didn't find anything, but some of the probes were bad. Some of the PCR reactions didn't work. And this led us to realize, when I saw this, I knew we were going to be able to do the map. And it then moved very quickly. Here's the team at the time. The 2 of us, Buddy Brownstein, who was running the screening team, and me, who was encouraging everyone, and these are the people doing the work. There was one final step in facilitating the map, and that was the development of a clever technique by Juha Kere, a postdoc in the lab. He found a way to isolate the ends of a YAC, and then we could sequence those, make a PCR reaction, and look for the next YAC so we could walk the furthest possible distance from the YACs we already had. That accelerates things very greatly. If you do simulations, this is really a big advantage in assembling a map. This was the paper on the walking with the sequence tagged sites from the end fragments. And within a short time, Randy Little, a graduate student, and Giuseppe Pilia, a terrific postdoc, had a map which spanned a full chromosome band for the first time. So over 8 million base pairs, 10 to 15 centimorgans, with complete continuity and totally formatted with all the genes in the region, including the ones that were known and new ones. This contains factor 9, other genes that are medically interesting. We actually then turned toward long-range sequence analysis to show that we could get there. And we took 219 kb of very high GC-DNA. This is the highest GC-DNA in the genome, so it's the hardest to sequence. We showed we could get contiguous sequence with our friend Elson Chen's laboratory. Here's a bit of that map. It's quite complete, and it's actually the primitive version of the genome browsers that we use now, showing all kinds of features of where things fall and exons and introns and so on. This was actually the longest sequence of contiguous human DNA for about 3 years before the sequencing centers really got started. So we reached the final chromosome YAC STS map. Nagaraj, my long-term colleague, assembled it. It has 75 kb STS resolution, so that means 2,500 STSs. And it revealed all kinds of features of the chromosome that had not been known. It's interesting that the total length that Nagaraj reached, 165 megabases, is right on to the final sequence length. And the positioning is also quite good. So all the inferred lengths and distances were later confirmed. It's a little bit of the map to show you the extensive amount of work that was involved and the amount that was then available for use. The next phase, you should all know, even if you're very young, that while these 2 chromosomes were being done by our center, a number of other centers gave partial maps of other chromosomes. So there were a lot of landmarks available, and those could supplement the sequencing centers. So the rest of the genome work was essentially done by sequencing centers, including prominently the one at Washington University that was taken on by Bob Waterston. The clone maps had a special value apart from the sequence, and that is that they were instrumental in finding disease genes. This was a big boost for the Genome Project at the time. The first one found was the Fragile X gene with the triplet expansion, which turns out to be very interesting in a number of human genetic conditions. Then anhydrotic ectodermal dysplasia in our lab, and then by Uakere, and then Giuseppe Paglia finding the gigantism syndrome on the X chromosome, Simpson-Galabi-Bemol. So everything began to move faster. These combinations of clone-based searches and increasingly sequencing have, of course, continued to resolve the basis of diseases. SQ28 is especially full because this GC-rich region is packed with small genes which are involved in a large number of genetic diseases. I want to point out that the YAC clones remain the only existing source of large genes isolated intact in genomic form. So for example, there are very large clones containing the complete factor VIII gene. This one is 580 kb. There's a much larger one with factor IX. And those are just beginning to be re-examined and reused because they can facilitate a lot of studies. So you already have the sequence, but if you don't have the material, it's much harder to study big genes. The regulation of promoter and enhancers, because they're all included. The requirements to establish methylation, post-transcriptional control, post-translational protein modifications. Remember, these are human genes in intact form, so they're going to give you exactly what you would get in the human system. Even commercial synthesis of totally natural proteins becomes feasible. Now, let's go back. What about the ribosomal DNA that we started with? We still weren't there. Even the YAC cloning techniques that we had when we tried to clone large fragments of ribosomal DNA they would tend to delete out. And we kept going back to this. The last attempt we had made was 1985 when we made a yeast clone that lacked most of the known recombination systems by mutation. It still would recombine out the inserts if they had a number of tandem repeats. So we couldn't do it. Furthermore, at the time, and still now, I would say, everybody assumes that if you have a lot of copies of our DNA, they're identical to that unit that was assembled in 1993. So why study them? There are many repetitive regions within the rDNA locus that make the sequence hard to assemble. The clones that I'll show you, we've initially sequenced by Illumina HiSeq. The sequence assembly programs collapse if you try to assemble ribosomal DNA sequence from large clones in this with short reads from Illumina. And the megabase size clones are unstable. Finally, last year, the technology appeared. So we resumed the rDNA analysis with the resolution of the problems. And we did it with new technologies. A modern version of what's called transformation-associated cloning that I'll show you, and new sequencing technologies. The implemented strategy was as follows. We focused on ribosomal DNA from a single chromosome 21, which came from a Japanese donor. And in fact, the reference sequence that we've done for this, which is now being submitted, will be the first part of the human genome reference sequence that comes from a Japanese donor. We then sequenced many YAC clones that contain just 1 to 2.5 repeat units. With this cloning system, that avoids the instability problem. And that enabled us to begin the sequence. In order to do this, we had to have a collaborative effort of 3 NIH groups. The YAC cloning was done by Jun-Yun Kim and Vladimir Larionov's group at NCI. They developed the transformation-associated recombination system. And Adam Philippi and Alex Dilthey at the Genome Institute had put in use long-range PacBio and nanopore sequencing. For those of you who haven't seen this used before, the PacBio will give you very long reads that are quite accurate. Nanopore, we've been able to go easily the length of the clones that we had, so several hundred kb. And in experimental sequencing, you can go a megabase at a time. The sequencing is not as accurate, but it's enough to give you a framework to pull together shorter clones. And then the annotation, the analysis, which was done by Nagraj and me. At last, we get to some data. First, the starting material to show you a mouse-human hybrid cell line that contains the human chromosome DNA that we used, all the mouse chromosomes that are labeled with DAPI, and the single human chromosome here, which you can see specifically with a chromosome 21 alphoid sequence or a sequence from the ribosomal DNA. So that was the starting material. Here's the cloning route, very powerful route that starts with genomic DNA and what's called a TAR vector. This contains what you need to grow the artificial chromosome. And this can be now a human artificial chromosome. The hooks are any sequences that you want to put in from the target that you have. They can be either in opposed directions or in the same direction so that you can pick up a number of other regions. For example, I'll show you some that are palindromic, would only be recoverable with hooks in that orientation. So you capture the DNA by cone transformation in yeast. The efficiency of this has now been increased by using CRISPR techniques as well to make cuts at the appropriate place. You get a high yield of YACs, which we then transfer into E. coli as BACs, and we take the BAC DNAs and sequence them. Here are the actual clones in the first report that's just being submitted now. And these are the structure of the rDNA based on the sequence in a number of isolated clones from the chromosome 21 we started with. And you can see in these clones exactly what you might expect. There's an 18S, the 5.8S, and 28S, the mature ribosomal RNAs, and then the 30 kb of what's called non-transcribed spacer, which then leads, in the clones that are big enough, to another such unit. That makes sense. You're getting sequences that are what you expect. About a third of the clones, however, showed a stranger structure in which the guts of the ribosomal DNA had been deleted out. Each of these clones is different. They start at different points, but all of them then end up with an end-to-end cloning structure in the DNA, which again can then only be cloned by having parallel hooks that go in the same direction. Otherwise, we would never have seen these. Now, the basis for the diagrams that I'm showing you comes from the sequencing. But remember, the sequence is being made from a chromosome that's been hanging around in a mouse-human hybrid cell for many years. And then you clone it into yaks, and then you clone that into backs. How do you know that what you're doing is what was in the standard DNA? Well, this is one route for these strange structures of looking at original DNA, and this is the technique of fiberfish. So we can look along uncloned human DNA with in situ probes. And you can see that many of them show exactly what you expect on the chromosome 21, the sequence of the transcribed region and the non-transcribed spacer. But then you see these places where there are very strange, long non-transcribed spacers of different sizes, and those correspond to the odd structures that we had seen. I'll show you now a more direct demonstration of the sequence variants that we see and of these odd structures. So this is the critical slide of results. First of all, are ribosomal DNAs identical? No, they are not. We've seen in 4% of the ribosomal DNA of a single individual, 600 variants. There are actually a few more than that. There are exactly 600 right now, which would be 1 per 75 base pairs. In other words, the rate of variation that we see in overall human DNA. So all ribosomes are not created equal. There are many variants. If we look at the subparts of the ribosomal DNA, we find that there are widely distributed variants. There are many fewer in 18S and in half of 28S, but then the rest of the region tends to have very high levels of variants. And there are many in the 3' external transcribed spacers, some of which almost certainly affect transcription. This is in the transcribed unit. Here, the intergenic spacer has a huge yield as well. If we look at the variations types, there are many single base changes. There are single nucleotide and multiple nucleotide deletions, and there are single nucleotide and multiple nucleotide insertions that we see scattered around the entire rDNA unit. Both by Poisson and by direct demonstration, all the clones we've looked at so far are unique. We didn't clone anything twice because we were only at roughly 0.4 of the equivalent of mount on the chromosome 21, so you'd expect them all to be unique. Now, the most important feature of this slide is a validation. In the validation, we went directly to deep sequencing of individual complete DNAs. And we took 4 individuals, 2 East Asian and 2 European, and sequenced them in depth. And we asked, do we see the variants in uncloned DNA that we found in the clones? And this led to surprises. In fact, we see a large fraction of the variants that we saw in 4% of the rDNA of one individual in a number of the individuals who are deep sequenced. Up to 80%, for example, of the SNPs have been seen, and the analysis is incomplete. We then looked at RNA-seq from the same individuals. So now we're asking, are these variants in ribosomal RNA? And the answer is a very large fraction are. This is a surprise because one might expect variation, but what this says is that many of the variants, and this includes the odd structures, the palindromic structures, we could detect the junction fragments by PCR from uncloned DNA of individuals. They're deep-seated in evolution. They must have been there before the separation of East Asians and Europeans, which is greater than 40,000 years ago. So ribosomes are not homogeneous. There are subclasses of ribosomes. Here's the positions of some SNPs and indels, for example, in the 18S and 28S ribosomal RNA structure. And these can be assessed for potential functions in changing secondary structure. A number of them are very suggestive, though of course there's no proof yet that those are true. So the conclusions of this rDNA project, still not at the complete end of this experiment, but we know how to do it now. We could successfully sequence YAC clones using the new tools for sequencing, PacBio and Nanopore long reads. We've done about 0.8 megabases, so again, that's 4% of the rDNA of one individual. There are SNPs, insertions, and deletions across the region that already show variants that are similar in prevalence to other genomic DNA. And we validated a very large fraction of variants in deep sequence DNA from 4 individuals from 2 widely different ethnicities. So again, these are in populations from the start of human evolution. As always, with genome analyses, it's not the end, it's just the beginning, and a lot of new research is open now for ribosomal DNA. You could think of a lot more things, but just to point out, of course we can begin to look at the effect of variations in our DNA, either for traits in GWAS, because there will be some variants that are specific for individuals and changes in ratios of variants, and even for possible involvement in ribosomopathies, an odd group of genetic conditions which have been shown now to involve some ribosomal proteins and could be modulated or even speculatively may occur by changes in the rDNA. And there are lots of questions. Now we can begin to look more carefully at how rDNA transcription is regulated on rDNA variants that are different in their promoter regions. What are the enzymes involved in processing? None of them has ever been determined because no one was working in the field. How do ribosomes turn over? Do the numbers of copies of rDNA vary among individuals? If you heard the talk earlier this week by my associate Jun Ding, that's an active field for mitochondrial DNA. And the programs he's developed with that are directly applicable to ribosomal DNA as well. This is a very interesting and odd question. How are the sequences of the ribosomal DNA on 5 different chromosomes coordinated in concerted evolution? Presumably, a variant originally arose on one of the chromosomes, but we see them on all the chromosomes from the frequencies that we get from deep clone DNA. And that's a problem. The only way that we know that you could ordinarily put together keep going with a whole bunch of things on different sites on a chromosome is by recombination and some kinds of rectification mechanism. But there's never been an observation of acrocentric chromosomes coming together. And what would be the telltale for human geneticists would be Robertsonian translocation. They're very, very rare. So that represents also a new field and a new question. Many opportunities for the future, and I hope perhaps some of you will be able to resolve these questions yourselves. And thank you for your attention.
Okay, so thank you very much for the very nice and wonderful lecture. So, my name is Haruhiko Shiomi, head of Department of Molecular Biology. So, I've been studying RNA and its binding proteins, including FMR1, which is the gene responsible for Fragile X syndrome. So, I am very much interested in post-transcriptional regulation of gene expression, including translation, messenger RNA stability, messenger RNA transport and localization, and splicing, and small RNA-mediated silencing, among others. So his talk was very educational and reminds me of many histories and many things. Thank you very much. So now I would encourage young people to ask many questions and to make a good interaction with David. So please. So you first.
Yeah.
Thank you so much for amazing talk. I'm quite impressed. So I was wondering that I'm quite surprised that many ribosomes have different sequences. So I was wondering how those mutation or change of nucleotide affect ribosome function eventually.
Well, that's the next phase of the work, clearly. I think they don't occur, and this is again a question of how that's prevented. They don't occur in the core structure that's involved in the actual formation of the peptide bond. So they're in these evolutionary expansion sequences. But those are important, of course, for the structure, perhaps for the rate of movement. There are all kinds of questions that come up. There's just no information. I mean, you have now seen the finding that there's variation in ribosomal RNA and DNA. How that affects function is of course, an open question. I wish we knew. It'll be interesting to see. If we've gone on too long, we can talk after the after the talk. Yes.
Sure. So maybe I missed some part. But, do you think as we got old, maybe we can accumulate some substitution of nucleotides in some copy of ribosome DNA, and which will be related to some aging disease or whatever?
It's possible. The variants that we're talking about are largely there in everybody, so they'd be there in young people as well. One of the earliest theories of aging, there are many theories of aging. You'll see some of them tomorrow if you come to the lecture then. That's the secrets of the nature of aging tomorrow. But the earliest theory was by Bernard Strahler in Toronto, and he suggested that what happens during aging is the loss of ribosomal DNA units, and that this then leads to the slowing of growth and the inadequate amount of metabolism. In fact, no one has looked. It's something that we're actively doing now. But in yeast, there's a long history of this, where rDNA circles form with aging and are associated with the aging phenomenon. It's hard to look for circular rDNAs in mammalian cells because they'd be very large, and that's hard to enter gels and so on. But it's possible. So it's not clear. Copy number might vary. There's no reason why there couldn't be mutations as there are. That's the mitochondrial theory of aging, that mutations accumulate with age. None of these theories have really been adequately tested, much less proven.
I see.
Okay.
And also related to what you explained to us about the, it's like a cutting edge technology and the science thing about the genome. So right now probably you are facing with, maybe you need more, it's like a revolution in the technology right now, right? Because to see the deep inside of the ribosome DNA probably. Do you have any idea about what kind of technology you need to accelerate your asking your questions?
Well, you could do something with cryo-electron microscopy to look at whether there's any gross formation change. The functional tests are possible. Some of Harry Noller's work suggests particular steps in ribosome function that could be tested. Since we don't know the details of processing or silencing of ribosomal DNA copies, it's very hard to know how we would test further about them. But I think biochemical tests should be possible. If it doesn't affect anything biochemically, it's unlikely to be important for translational research. But it would be surprising if all of these changes are carried along through evolution and are not functional.
Right.
At least some of them are probably doing something, or they would have been lost.
Yeah.
Or selected against, as they are in 18S.
Right.
Whether they're actually subclasses of ribosomes is not clear. You might find that some are specialized for certain classes of message. Almost anything is speculatively possible at this point.
Yeah, very thrilling. Thank you very much.
Yeah.
So any further questions?
Yeah, it's
Yes,
late.
please.
It's really getting late.
Thank you for a nice talk. I want to ask you, around half of ribosomal DNA gene transcribed in human cells among 400 copies of ribosomal DNA. And you, in your presentation, you showed there are many variant ribosomal DNA locus. So this variant may affect which ribosomal DNA locus is transcribed. Do you have any?
Yes, well, that's, you want the next experiment.
Yeah.
I'm glad that you do, but we don't have any information about it at this point. Again, this field has been dead practically for 25, 30 years. And now it's time for it to wake up, in my opinion. Okay?
Any questions? Yes.
When I was in undergraduate student, I was doing a chromosomal fish, and I remember that, and I did many ribosomal RNA genes, so it's very nice talk. And I remember that there's another ribosomal RNA cluster, 5S. You didn't talk about it.
We studied that. There's a paper from two of my students in 1989, which is the paper in the field. And as many papers in my lab, I'm not an author. They looked at the 5S cluster. It's on chromosome 1, Q41. So all the 5S DNA units are there. If you treat with a restriction enzyme that's only present once in the repeat unit, it all collapses to a 2.2 kb unit. The number of copies varies among individuals. We saw that. But that's the only paper in the field. Nobody has gone back to ask whether it varies in structure. My guess is it'll be completely homogeneous, because that's a single locus, so you can have gene correction. Okay.
And my question is that how do you think about why the 5S is separated from other ribosomal RNAs?
In many lower organisms, it is part of the unit. As you go up to later evolution, it separates. I guess the important thing is that there's enough of it to saturate what's needed for the ribosome. And so it just went off on its own. But nobody was there to see what happened. So it's not clear what the event was, or obviously it's not fatal, fortunately.
Thank you.
Okay.
Okay. So if no more questions, so I would like to close this wonderful lecture. Thank you very much. So.